Hello everyone. First of all, a couple of words about myself. My name is Mustafa. I'm Turkish of Arabic origins. I joined Dr. Edward Porta's group in Spain, in Barcelona, a couple of years ago. And I'm in my second year as a PhD student. And I've been working on this exciting project titled The Spatial Landscape of Cancer Hallmarks Reveals Patterns of Tumor Ecology, which is published as a preprint as of now. And I'm gonna go ahead and start telling you about it. So first of all, a quick background to the motivations of this project. As you very well know, if you're in the cancer field, that tumors are much like ecosystems. They exhibit multi-layered molecular alterations that could be driven by different factors, such as the cell of origin, the biological sex, the germline background, and the age of the patient. All of these factors contribute to the complexity of cancer ecosystems. And two leading pioneering scientists in the field, Hanahan and Weinberg, came up with this beautiful, elegant framework that simplifies this complexity, which they called Cancer Hallmarks. For instance, one of the earlier versions of this concept suggested that cancer cells have the ability to evade growth suppressors, or they have the ability to instigate genomic instability and undergo mutations. And one of the latest hallmarks that were proposed is the fact that cells can undergo senescence, or that they can undergo epigenetic reprogramming, and so on and so forth. And in our group, we are very much interested in understanding how these hallmarks are distributed throughout the space of a tumor. And to do so, we use a state-of-the-art technology, which is called the Visium Spatial Gene Expression Protocol from 10x Genomics. And essentially, it's a slide with 4 capture areas. Each capture area is a 6.5 by 6.5 millimeter. And in each of these capture areas, you have barcodes, spots where there are millions of barcoded RNA molecules that would bind on that would take the RNA from the tissue that you put on the slide, so that there is hybridization, and then you can sequence your slide, your section of interest, and then you get in the end a matrix of gene expression where you have the columns representing the location of these reads in the space of a tumor. And this is quite important, especially in the current era of spatial omics, and it addresses quite a varied and interesting, broad questions that is relevant to cancer biology. And so in this particular study, we are using a pan cancer cohort of primary untreated tumors from 10 cancer types. At least having 5 per type, and maximum we have 9, and totally it's 63 samples as of now. And what I'd like to show you is a glimpse of the pipeline that we took here, and it starts with a very important and critical step in which we assign particular gene sets to each hallmark. We've invested quite a lot of time in manually curating gene sets for each of the 13 of the hallmarks proposed by Hanahan and Weinberg, and we go to the Pathway Commons database. We, for instance, I'll give you an example of non-mutational epigenetic reprogramming, where we have pathways related to chromatin remodeling, histone acetylation, deacetylation, methylation, or demethylation. And in the end, we end up doing a very similar approach across the other hallmarks. And we have a set of genes, about 2,600 unique genes. Each hallmark has about at least 100 genes. That varies up to maybe 600 genes. And in the end, we map these gene signatures on the tissue section that we get after the wet lab protocol. And here in the middle, I show you an example of a breast adenocarcinoma, and at the bottom of it, you see the signature of non-mutational epigenetic reprogramming quantified on that tissue section. And on the right side is another example of colorectal adenocarcinoma, and these top slides are hematoxylin and iosine staining, by the way. So at the bottom of the colorectal slide, you see the activity of avoiding immune destruction quantified on that tissue slide. Now, I'd like to show you that, as you can see, these signatures visually, they look spatially clustered, right? And in order to quantify this phenomena, we use what's called Moran's I, which is a spatial autocorrelation metric. Essentially, if it gives you a score of 0, your feature of interest, in this case, the hallmark signature, is randomly distributed. Whereas if it's closer to 1, it means that your feature of interest is spatially clustered. And if it goes to -1, which can in some cases, which we don't see in our case, if it goes to -1, it means like it's more like a repelling, that features are repelling each other throughout the space. So in our case, you can see that the hallmark features across all cancer types here are grouped together on the right-hand side. They have an average of 0.8 Moran's I, indicating they are very highly spatially organized. And so we came up with a couple hypotheses. We asked, is it the cell identity that contributes to this organization? Is it the cancer clones? Is it the spatial dependence? And first and foremost, we needed a way at the beginning of this project to quantify where these, to quantify where the cancer component is and where the microenvironment compartment is. And we used a classic method, which is called estimate, which has been used a lot in TCGA studies. It's been used successfully. We've applied it on all of our samples, validated by pathologists on 17 of these samples from 5 cancer types. Essentially, on one side of the score, it gives you a proxy to the neoplastic content, and on the other side of the score, it gives you a proxy to the TME content. And so after having these clusters, we quantify the contributions of these compartments, the neoplastic versus the TME, to the signatures of the hallmarks that we developed. And what we find is a nice pan-cancer architecture whereby a set of hallmarks appear to be more relatively active in the tumor microenvironment, whereas another set of hallmarks appear to be more relatively active in the cancer compartment. And this clustering is notably not driven by the source, whether or not it was collected from a public or an in-house data set, nor is it driven by the tumor type or the tissue embedding method. And from here on, we noticed that even though the cell identity does explain to a high degree this spatial organization, what led us to explore the second hypothesis, which is the cancer clones, was the observation that a cancer hallmark whose activity is high in the neoplastic compartment could exhibit a wide variety of activity throughout the space of the neoplastic compartment itself, as an example, which you can see here for enabling or replicative mortality in a kidney cancer sample. And so to explore this hypothesis, we used infer-CNV, a method that is used usually on single cell data, but it has been used also on spatial in several published papers already. And the main question here is, can we actually find a correlation between genomic distance between the subclones and the difference in hallmark activity, that is the difference in the phenotypes? Do they correlate? And first of all, on the left-hand side, the bar plot shows you the distribution of the genomic distance. On the x-axis is the genomic distance. So the higher it is, the more genomically distant pairs of subclones are. So the y-axis represents the number of the pairs of subclones. And you can see that if you split it you can make a threshold where we say that less than 10% genomically distant pairs of clones can be called genomically similar, or there's low distance. And if you take those that are higher than 10%, we would call them these subclones are genomically distant. There is a higher genomic distance. And on the right-hand side, what we do is we take the hallmark difference between the subclones on the y-axis, and we split these differences by those two categories that we defined in the plot on the left-hand side. And what we see is a nice general correlation, indeed, between the genomic distance and the hallmark difference across the different cancer hallmarks that are more relatively active in the neoplastic compartment. However, you can see that these box plots have quite interesting variations. And that tells us that this is not a perfect correlation. In fact, the average correlation was about 0.4 across all of these neoplastic hallmarks. And so, yes, cancer clones does explain this spatial organization within the neoplastic compartment to some degree. However, we also know from a growing body of evidence that cellular crosstalk contributes to the functional determination of the tumor itself. And so there is definitely some importance that should be given to the spatial localization. And this is how we leverage the technology here to address this question by using a machine learning approach, whereby I give you an example here where we want to predict the spatial distribution of a hallmark that is more active in the tumor microenvironment. If you look at the right-hand side, for example, you have a set of 6 hallmarks, right? Which we call the TME hallmarks, those that are active more than TME. And on the left-hand side, you have a list of 7 neoplastic hallmarks that are stitched together, more like stitched together. And so here the idea is, if you take the green hallmark, which is sustaining proliferation, and we want to predict its activity throughout the tumor microenvironment, what we do is a very nice trick. We take the activity of the predictor, which is on the left-hand side. I show you here an example of non-mutational epigenetic reprogramming, which is mostly active in the neoplastic compartment. But in order for this hallmark to be able to predict the activity of another hallmark, which is the green one in the TME, we have to translate its activity somehow into the TME compartment. And to do so, we develop what's called a radar score, which is a very simple approach. A radar, as the name suggests, is like putting a radar in the tumor microenvironment so that it detects how much activity of non-mutational epigenetic reprogramming is happening from the cancer compartment, weighted by the distance of each of these neoplastic spots to where the green hallmark is active. So essentially, you are putting radars in the compartment where you want to predict those target hallmarks, in this case, the tumor microenvironment hallmarks. And so when we do this experiment for all of the TME hallmarks, and then we switch the experiment to predict all of the neoplastic hallmarks using the TME hallmarks together as predictors, we see that the random forest model gives us a very high predictive power, predictive accuracy, in which case you see the R squared of these models is on average 0.7, more or less, across all of the hallmarks. And the end of it is what you see on the right-hand side. As an example, I show you a kidney cancer tissue slice where the model told us that avoiding immune destruction activity in the microenvironment, which is the TME spots, which you see on the blue colored compartment, whose maximum activity is in light blue, is the best predictor, or one of the best predictors, of genomic instability and mutations that is occurring within the neoplastic compartment, whose maximum activity is shown in bright red. So you see nicely how the bright red and the bright green are very much spatially proximal to each other. And this is why the model told us they are very highly predictive. Now note, this high predictive power could be either by spatial proximity or spatial distance. And this is the really interesting part that I want to show you here is when you summarize what we call the spatial dependency, meaning whether or not they are spatially proximal or spatially distant. In this example, where we predict the hallmarks of the microenvironment using the hallmarks of the neoplastic compartment, on the y-axis in this heat map is the predictors, which are the ones from the neoplastic compartment, and on the x-axis are the target hallmarks that are in the microenvironment. And I highlight in this black box an example of the first hallmark, evading growth suppressors. It has a very strong spatial dependency with the black hallmark, which is activating invasion and metastasis, but it has at the same time very negative spatial dependency with enabling replicative immortality, and genome instability and mutations, the bluish hallmarks. What, in our interpretation, this could mean that cancer cells who are fit to metastasize could be more capable of evading growth suppressor rather than having genomic instability or replicative immortality. So this is one of the observations that we see across all of these experiments that we have been running, and due to the limitation in the time, I'm unable to go through much more than that. But in the preprint, you will see much more examples of such observations that we have found through leveraging the spatial information in the analysis of the hallmarks together. And so in the end, we do find a high explanation of the spatial organization of the hallmarks through the spatial dependence as well. In the end, we have a working model on a pan-cancer level where different hallmarks from the neoplastic compartment could be distributed depending on their relative distance from the TNB compartment and vice versa. And this model, of course, can change depending on the cancer type, could also change depending on the sensitivity to different treatments, whether or not patients are responders to treatments or not. So this model is preliminary, it's pan-cancer, but it could really change depending on the cancer type. And finally, we do believe that it has a translational potential, where on the left-hand side, you see what we call a hallmark map, where on a tissue slice, you could map the activity in each spot. You could map the activity of the different hallmarks and see their map for any particular cancer type of interest. And this could be integrated with histopathological annotations from single markers, such as the EpCAM, the keratins, and so on, integrated all together with the hallmark map in histopathological settings. And finally, I'd like to thank my group leader, Dr. Edward Porta, and my co-author, Sergi Servia, the co-first authors, Sergi Servia and Daniela Gracias, and the rest of the team. And I'd like to thank the collaborators and the funding agencies, and particularly the La Caixa Foundation, for giving me for granting me the opportunity to do this project. And thank you all for listening. I'm happy to take your questions.